How we diagnose the malaria? We diagnose the malaria like any other parasite, direct, indirect, and molecular. In the direct, we have at first the peripheral blood examination. The peripheral blood examination still until now is the gold standard. Up till now is the gold standard. The blood examination, the blood film is stained with Giemsa. We have a thin blood film and thick blood film. The thick blood film, or we call it a thick drop. The thick drop is used for concentration. By the thick drop, I can tell that this patient have malaria only. The thin blood film is done to verify the species, to identify the species. So you have to do both tests, the concentration test or the thick drop and the thin drop. The thick drop, I can tell that this patient you have malaria. But for the treatment, I have to know the species. So I have to do the thin blood film. The thin blood film to identify the species, the morphology of the parasite. So I can tell this falciparum have a special drug, special treatment. For the vivax and oval, another treatment, and so on. So this is the direct method, the first thing. So in the direct method, we have peripheral blood, we have the quantitative Buffy-Cope technique, or we called it the QBC. It is very rapid, it is sensitive, it is specific, but cannot diagnose the species. This is a centrifuge tube, but it is a QBC tube, usually it is very thin. It is coated by what is called as acridine orange. This acridine orange is a fluorescent dye. Then the blood sample is collected and it is centrifuged. When it is centrifuged, we have three layers. We have plasma, we have Buffy coat, and we have the red blood cells. The parasite is present inside the red blood cells. They examined by the fluorescent microscope, so we can tell that there is a malarial parasite. But we cannot identify the species. So it is rapid, it is sensitive, it is very easy to do, but it cannot identify the species. The third method in the direct is the sternal puncture. So usually in the direct method, we have a blood film, a thick drop. It is a concentration that there is a malaria, a thin blood film, and the thin blood film is to identify the species. The second method is the QBC, which is a very simple method, very rapid, very sensitive, but it cannot identify the species. And it can even be used in other parasites. And the sternal puncture. The stages, as I told you, detected in the peripheral blood, all the stages are detected in Plasmodium, Vivax, Oval, and Malaria, and in Falciparum only the gametocyte and the ring are present. The trophozoite and the schizomes are present only in fetal conditions because they are present inside the capillaries. This is the shape of the parasite again. The ring stage in Vivax, it is 1/3, 1/3, 1/3. The red blood cell is enlarged and oval. Here is normal in size. Here multiple infection. It may contain more than one chromatin dot and the ring. Then the trophozoite of the Plasmodium. We have the Vivax, which is present in the enlarged red cell. It is amoeboid in shape and contain a vacuole. Amoeboid and contain a vacuole. The red cell is enlarged and fimbriated. Here is more evident than the previous slide. It contains a vacuole also. It is amoeboid, contain a vacuole, but it is compact. In Malaria, it is sometimes contained, is compact and band-shaped, and it contains the hemozoin pigment. Here are very evident. The hemozoin pigment are evident here. This is not seen. The trophozoite of falciparum is not seen in the peripheral blood. The schizont of Plasmodium, it depends on the number of merozoites. The number of merozoites in vivax from 16 to 24 or to 20, increased number of merozoites. Here from 6 to 12 and 6 to 12, but the difference between the oval and the Malaria, the red cell here is enlarged and oval. Here it is in the form of rosette shaped. In the falciparum, although it is compact, it is not seen in the peripheral blood. The gametocyte of the three species, the gametocyte of Plasmodium vivax is round. It contains a chromatin dot. Also, the pigments appear here, oval, fimbriated, and chromatin dot in oval. And the red cell is enlarged. The red cell of normal size, it is smaller in size and compact, but in falciparum it is in the form of banana shaped or crescent shape. This is a blood film for Plasmodium vivax, where I can see the schizon, I can see the ring stage, and I can see the trophozoite, and I can see the schizon, trophozoite, and gametocyte. All stages are present here. This is another form of Plasmodium falciparum, where I can see the gametocyte and ring stage only in the Plasmodium falciparum. In Plasmodium knowleski, the RBCs are not enlarged, the pigments are spread, there is a pigments which are spread, more or less it has the shape, the trophozoite more or less, sometimes it is band shaped, and it is different in, it is similar to malaria, to Plasmodium malaria. Similar to Plasmodium malaria, it contains a high parasitemia like Plasmodium falciparum, but it is not as serious. And the attack occur every day. It is quotidian. The attack occur every day. It is not present in Egypt. It is in Southeast Asia. All this is for the direct method, the blood film, the sternal puncture, and the QBC. The blood film, thick drop and thin film, up till now is the gold standard. But we have the indirect method or the immunological method. The immunological method is either detection of antibodies or antigen. Detection of antibodies in indirect hemagglutination, ELISA, immunofluorescent, these can be done only in blood donors to prevent the spread of malaria to recipient. So the immunological test, the detection of antibodies, is done only to the patient which are a blood donor, to be sure that they have not malaria and they will not transmit the malaria to the recipient. The immunological test which is important is the detection of the antigen. It is more important than the antibody and more diagnostic is the detection of the parasite antigen. It is the immunodiagnostic dipstick test, the immunodiagnostic dipstick test. It is for detection of the parasite antigen. There is a nitrocellulose paper which is soaked with monoclonal antibodies and a colored dye, then a blood of the patient's blood. If it contains the parasite antigen, mainly histidine-rich protein in falciparum, it is positive. If not, it is negative. So the immunodiagnostic test or the dipstick test is important for the diagnosis of Plasmodium, falciparum, and it denotes active infection. It denotes active infection. So the blood film is very important for diagnosis, and the immunodiagnostic is important for the diagnosis of active infection. And it is for detection of antigen. the detection of antibodies, it is only for the blood donors. So blood film, indirect. The third method is the molecular biological method, or the polymerase chain reaction PCR. It is for the detection of the DNA of the parasite. There are other laboratory techniques. Beside the hemolytic anemia, there is a normocytic normochromic anemia. The platelets are usually normal. The fibrinogen is elevated and the platelets are reduced. The platelets are reduced, but the fibrinogen are elevated. The bilirubin and the transaminase concentration are raised because the liver is affected. So when you think about the laboratory findings, you have to think about the pathology and the clinical manifestation. There is affection of the liver, so there is elevation of the bilirubin and the transaminase. And in case of quinine treatment, there is a hypoglycemia. These are other laboratory findings. Before ending my lecture, I want to tell you that there is a difference between the schizont, the hepatic schizont, and between the hepatic schizont and between the erythrocytic schizont. The hepatic schizont contain thousands of merozoites, but the erythrocytic schizonts contain tens, 10, 20, 12, 16, and so on. The hepatic schizont doesn't contain pigment because this parasite is pigment-producing parasite in the red blood cell only. So it is present only in the erythrocytic schizont. So no need for the pigment. So the hepatic schizont. Number 3 is the presence of hypnozoites, and they are present only in vivex and ovale, and they are not present in the other stages. Okay? And by the laboratory diagnosis, I can end my lecture, and thank you very much for listening and for your attention. Thank you very much.